Continuation of the video of cell flagella functioning. Flagella has three proteins: tubulin, nexin, and dynein. Tubulin is structural protein, and nexin and dynein are functional protein. The functioning is done by due to the sliding of doublets. These are the two doublets, these two pores, which are connected by the nexin protein, that is red. And when one of them is stretched, okay, if this doublet is if any part of this doublet is stretched, then the nexin protein also gets stretched. And due to this and if the doublet is stretched, it keeps stretching on and returns back into its normal position. And that is exactly how flagella works. This is flagella, and due to the stretching of nexin, the whole structure molds in some way and helps in the movement of water or anything else. Now, if the movement gets to the constricted form, then it is called power stress, and if it returns back to its normal position, then it is called recovery stroke. Now, nucleus. It is the seat of genetic information of cell. It was discovered by Robert Brown. Number of nuclei depend, and so it names change. If one is present, then it is called uninucleate. Two, binucleate. Example: paramecium has macronucleus and micronucleus. If many nucleus are present, then multinucleated cell. Multinucleated cells are again of two names: cenocellic and syncytial. Syncytial is used for multinucleated animal cells, and syncytial is used syncytial is used for the fungi in plants. Example: rhizopus, eukarya, and coconut milk has liquid endosperm, which has many nuclei. Okay, the liquid is many nuclei, and the endosperm is the storage of in the seeds of monocots. Enucleated are those which nucleus are lost. Example: mature RBC and sieve tube. Shape: most common shape is spherical shape. Kidney shape or bean shape or reniform, all three are same. Is the shape of, example, macronucleus of paramecium. This is kidney shaped, this red one. Now, red lobed shape is WBC. Structure of nuclear envelope, nucleoplasm, that is inside of it, and nucleolus and chromatin reticulum. This is RER. These are the ribosomes, the red ones: outer membrane, inner membrane, and the chromatic reticulum, which are light colored and dark colored both. And then there are nuclear pores where nothing is present. Okay, connecting link. Now, nucleolus is present, and then there are nuclear lamella attached to the inner membrane. Now, nuclear envelope, the first part is double membrane, and outer membrane is associated with ribosome 60S subunit. We know that Christine of endoplasmic reticulum is that only. Now, outer membrane is continued by RER, so it is appearing rough. And perinuclear space, that is space between outer and inner membrane, is filled with matrix having very few enzymes. Inner membrane is associated with nuclear lamina. Nuclear lamina are the proteinaceous filaments. They provide support to nuclear membrane and maintain the shape, acting as a site of bonding free of DNA. And DNA will separate them from the nuclear lamina only at the time of cell division. Nuclear envelope is not complete but has perforations, means nuclear pores. These nuclear pores are for exchange between nucleus and cytoplasm. Now, if you see here, there is diagram of this. It is ball like, nucleus is ball like, sphere like. It is not two dimensional. So if you see the surface of the it is, then you will find ribosomes which are attached only at the RER extensions of it, and also on the outer membrane. And you will also see some places, at some places there would be um, nuclear pores. That would be if it is, think it like a 3D shape, it's a sphere. So you would see that this would be surface from one side, and outside you would see many nuclear pores, which are nothing but pores for transportation. Now, nuclear envelope transports the following: enzymes, proteins, nucleotides, ATP or GTP, and mineral ions. These are transported present in the cytoplasm. And RNA, that is mRNA and tRNA, ribosomes units, are from the nucleus. These seven things are transported. Regulation of nuclear pores. Number one, electron dense plug. Three types of nuclear pores, or where the nuclear pores can be found, are there. Now, first is electron dense plug. The second is septum. Okay, it's electron dense plug. There is nothing but density of matrix is very high, so only small particles are allowed to pass through it. And then septum is a highly specialized membrane which is highly selectively permeable. Okay, very highly selective. And it also helps in the regulation of nuclear pores. Again, the uh, type is third Third is pore complex. It is highly specialized. It has a nucleus called structures which have spokes depicted regulated by contraction or relaxation of spokes. As the spokes contract or relax, so does the method of transportation across it depends. Now, substances can also be passed through bulk transport, that is the cycle method, without using nuclear force, which is this. Simply vesicles come here, attach to the outer membrane, detach from the inner membrane and go inside the nucleus. Now, nucleoplasm. Nucleoplasm is a proteinaceous protein matrix or round substance of nucleus, comparatively acidic in nature. Acidic due to proteins of itself. It has their own proteins. It is non-staining part of the nucleus because nuclear stains such as acetocarmine do not stain the nucleus. Nucleoplasm, it stores no functions. It helps in the integrity of nucleus, acts as a medium for functioning of DNA. It also stores molecules like enzyme, protein, mineral ions. Nucleoplasm will be the functional base for functioning of DNA for nuclear functions, that is replication and transcription. Nucleolus is called as little. Now, third part, nucleolus. It is called as little nucleolus because it appears as a dark spherical spot inside the nucleus, resembling nucleus in a cell. It is spherical in shape but no limiting membrane. So, the shape is maintained by calcium ions. Perfect spherical shape. Parts of nucleus. Parsomorpha. Parsomorpha, that is amorphous matrix made up of proteins. Then fibrillose, that is fibrillar region. And then pars granula, which are granules present in this. Okay, and lastly nuclear chromatin, which is nothing but simply DNA part that goes through it. This black one. Now next, here we have DNA does the transcription to form RNA. RNA has three rRNAs, 18S, 28S, and 5.8S. 5S RNA comes from outside of the nucleus from the cytoplasm. rRNA, these three combine to form proteins to form ribosomal subunits. These proteins form ribosomal subunits. These are not function inside this because we know that they are attached to each other in the cytoplasm by the magnesium ions. So, and ribosomes, that was first observed in nucleolus by Sao and Sato first found ribosomes in nucleus. Now, fourth part, chromatin reticulum is a network of colored threads spread in the nucleus. It is not uniformly colored or staining. It is of two types, euchromatin and heterochromatin. Euchromatin is the lightly stained part. It is the darkly stained part. Euchromatin is loosely coiled, has functional genes, undergoes transcription, and the first to start replication. Heterochromatin is dark stained part and it is tightly coiled. That is why it's so tightly coiled, and that appears dark and non-functional part. And since it is tightly coiled, so it means that it won't be frequently used. It's tightly packed up, and these are loosely coiled, so it means very regularly being used. These are, so they are functional genes, but these are not functional genes. Now no transcription occurs, therefore replicates later. Here, this is the first one to start replication. Now heterochromatin again of two types. constitutive heterochromatin, that is junk DNA, because not because it is useless DNA, it is very useful DNA, but it has no genes, so called junk DNA. These are two genes, let 1 and 2, and here is junk DNA between this. Okay, uh, here are two DNAs, 1 and 2, and here is junk DNA inside this, but it has no genes. Role, very important roles. It acts as a spacer gap between the genes, that is spacing between two genes. Now you would think, why is it that, or even a row, a change or mutation in one gene does not allow to affect the other genes, as gene of one DNA and other DNA are not connected. Okay, because of this, so mutation does not occur. That is how it helps. Now other part is that it acts as an absorber mutation, or any mutation in the junk DNA is not expressed, even if itself gets something mutated, then it won't let the others to be affected due to itself, because it never expresses its genes, it never expresses. Okay, and okay, chromosome will be condensed form of DNA, so that every gene will have a fixed gene locus. See, these are C band. Okay, and then homologous chromosomes are those chromosomes having same gene loci. Homologous chromosomes identify each other by their matching C bands. Now facultative heterochromatin, if needed, then this part of DNA is acting as a chromatin. They are genes which are not functioning in cell due to its differentiation. It is temporarily inactive, but temporarily, not forever. Now, in example, in cells of stomach, only some genes express. Other genes of insulin, salivary amylase, thyroxine won't express, as they are not part of the cell function.